MHC class 1 molecules bind short peptides of 8 to 10 amino acids by both ends. The binding of a peptide in the peptide binding cleft of an MHC class 1 molecule is stabilized at both ends by contacts between atoms in the free amino and carboxy termini of the peptide and invariant sites that are found at each end of the cleft of all MHC class 1 molecules. These contacts are thought to be the main stabilizing contacts for peptide MHC class 1 complexes because synthetic peptide analogs lacking terminal amino and carboxyl groups fail to bind stably to MHC class 1 molecules. Other residues in the peptide serve as additional anchors. Peptides that bind to MHC class 1 molecules are usually 8 to 10 amino acids long. The peptide lies in an elongated conformation along the groove. Variations in peptide length appear to be accommodated, in most cases, by a kinking in the peptide backbone. However, two examples of MHC class 1 molecules where the peptide is able to extend out of the groove at the carboxy terminus suggest that some length variation may also be accommodated in this way. These interactions give all MHC class 1 molecules their broad peptide binding specificity. In addition, MHC molecules are highly polymorphic. There are hundreds of different versions or alleles of the MHC class 1 genes in the human population as a whole, and each individual carries only a small selection of them. The main differences between the allelic MHC variants are found at certain sites in the peptide binding cleft, resulting in different amino acids in key peptide interaction sites in the different MHC variants. The consequence of this is that the different MHC variants preferentially bind different peptides. The peptides that can bind to a given MHC variant have the same or very similar amino acid residues at 2 or 3 particular positions along the peptide sequence. The amino acid side chains at these positions insert into pockets in the MHC molecule that are lined by the polymorphic amino acids. Because the binding of these side chains anchors the peptide to the MHC molecule, the peptide residues involved have been called anchor residues. Both the position and identity of these anchor residues can vary depending on the particular MHC class 1 variant that is binding the peptide. And that's MHC class 1 molecules bind short peptides of 8 to 10 amino acids by both ends. Have a great day.